to learn a little bit, we're going to complete the loop. We've learned about proteins. How does our body produce proteins? Proteins are so amazing. They have a wide range of functions. Okay? The proteins we have encountered so far. Okay, number one, we've encountered proteins, enzymes. Okay? Right now, membrane proteins, there's another class of proteins we encountered, right? Membrane transport proteins. Other proteins you can encounter are structural proteins. Structural proteins are proteins you can, for example, a keratin is a protein. It can be found in your hair. Another kind of protein you can find, proteins can also be hormones. They run through your blood, helping to control various parts of your body. Proteins are amazing. They have a wide range of functions. What determines their function? At the end of the day, what determines the function of a protein is its structure. We've learned that proteins have tertiary structure, secondary structure. At the base of it all, what is key is their primary structure. Because the primary structure dictates how the secondary and tertiary ones will form. Today we're going to complete this loop. How in the world does our body even create the primary structure of a protein? We learned that the primary structure is about the sequence, number, type of amino acids. How does our body create that sequence? Right? We sort of slightly touched about this because you're a curious class and you asked about this, I think, a few chapters before. But today we're going to complete that loop. Here is the DNA. What is in our DNA? Our DNA is nothing more than a recipe book. Okay, you don't have to write this down, but to me this is the best analogy I have. Okay, our DNA is like a recipe book. And our nucleus is nothing more than a central library where you can find a lot of the DNA. Yeah, I like to think of the nucleus as a library where you store all these recipe books, right, which is DNA. Yeah? Well, what are these recipes for? Well, our DNA contains the recipe to build any and every protein you can find within our body. Name it and we have it. If you want to build an enzyme, we have a recipe for that. If you want to build a hormone, we have a recipe for that. If you want to build a membrane transport protein, we will have that recipe inside our DNA to do just that. I'd like to introduce a new word today that you may not be new because you probably heard this a lot. We always say that it's in your genes. It is in your genes to be tall. It is in your genes to have black hair. It is in your genes to have brown eyes. Genes are nothing more than recipes. Okay? So if we ever say the word gene, a gene is nothing more than a sequence of DNA that codes for a protein. Okay? That's what a gene is. A lot of nucleotides together. Yeah? A gene is a sequence of DNA. So, sorry, when I say a sequence of DNA, I mean like a sequence, like this sequence over here. This could be a gene. It tells our body what protein, a particular protein that we can make. Every protein in our body has a gene for it. Or, if I put it in layman terms, every protein in our body has a recipe. We actually don't have the recipe in our DNA to make anything else. We don't have a recipe to make carbohydrates or lipids or nucleic acids, even though they are all very important. We only have the recipe to make proteins. Why? Okay, Olivier has phrased it quite well. Because proteins can do almost everything and anything. If you need carbohydrates, if you need lipids, why not just make an enzyme that can do that? So our cells and our DNA, very smart. We just choose one biomolecule that can cover all bases. They can do almost every single function. Our DNA just has the recipe to make all the proteins we ever require. And these proteins can build almost everything and anything else. Okay? nothing more than a sequence of DNA. When I say a sequence of DNA, I mean like a particular sequence of nucleotides. Yeah, sequence of nucleotides. But if you ever see people say sequence of DNA, it means that they're talking about a particular sequence of nucleotides. So for example, let's say here I have a gene on the whiteboard. I'm going to put dot 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 over here to remind us that our DNA is very, very long. Okay? Our DNA is extremely long. So let's say this is the DNA I'm looking at. That's really long. Maybe I'm just looking at this short segment over here. Okay? This short segment over here that you see on the whiteboard is just this part over here. Okay? So our DNA contains many genes. We have around, I forgot how many genes we have. Sorry? Okay, does genes have DNA or DNA have genes? What? What did I say? Our DNA contains many genes. Genes are sequences of DNA.
So then DNA
Right.
make up gene, right?
A sequence of DNA. Okay, let me think. Yeah, I think then therefore it will be more helpful for us. A gene is a sequence of. Okay, let's replace this nucleotides. Maybe that will be better for us. Because when I refer to DNA, I would say people use it quite loosely. They can refer to the entire molecule, or they can refer to just a sequence. So perhaps it will be more helpful for us at our level, sequence of nucleotides that codes for protein. But don't be surprised if you see a sequence of DNA. That's also okay in our phrasing.
So is a gene a DNA then?
A gene is a segment of the DNA, perhaps.
Then what's the other segment of DNA?
Right, okay, so our DNA is very long. Okay? Our DNA is very, very, very long. Genes are segments of the DNA that code for protein. Yeah, DNA is the bigger one. Okay, so gene is a sequence of nucleotides, or we can say it's a segment of DNA. Maybe this will fit our.
The other segments have other functions.
Sorry, yes, sorry.
So you like people have the same gene, make the same protein.
Right, okay. So if you have an eyeball, I have an eyeball, that means we must have the same gene to make the eyeball. Okay? So that's how we roll. Okay? And therefore if you see a cat has another part, another protein that we don't have, right, means that the cat has the gene to make that we don't have. Okay? What makes us human, right, if we look at each other, because we have the same genes.
Then what does it mean when like people say, oh you must have gotten this gene from your parents? Like what does that mean?
Right, okay, then if you think about it, it doesn't quite make much sense. What's the point or value of saying, I must have gotten this gene from my parent? If we all have the same gene, there's no value in saying this, right? Okay, so in GH4, you will learn that although we all have the same genes, we may have different forms of the same gene. What do I mean by that? For example, you have the gene to make a protein for brown eye color. I also have a gene for eye color, but it's a different version. It's for black eye color. So when someone says that you inherited this gene, what they're really saying is they've inherited this version of the gene from your parent. And actually you look at all of us, look at each other, we are all vastly different from each other. We have the same things, but slightly different. Yeah? Very likely, for example, look at our blood type. Right? You can even ask your partner next to you. Our blood types are very likely is quite different. But you know what determines blood type? It is the protein found on your red blood cells. Okay? It's the protein found in your red blood cells. So that determines. So we all have the gene, for example, for blood types, but we may have different forms of the gene, and therefore, we have different blood types. Can you see?
Then when you say version of gene, is it because like structural difference or like they switch out a nucleotide for another nucleotide?
Exactly what you just said. Okay, so this is all beyond our realm, okay. We will learn this in JH4. Okay, so FYI, but you will know this eventually. So when I say different versions of the same gene, right? What does that even mean? If this codes for, okay, notice the language I'm using. I always say a gene codes for. Okay, it can code for this protein, code for that protein, so on and so forth. If this particular sequence codes for, let's say, a black protein for eye color, then when I say that there's a different version of this gene, what do I actually mean? Okay, so this is just FYI, you will learn this in JH4. When I say different version of the same gene, it means that largely the sequence is the same. That's why we still say it's a gene. But there can be slight differences in the sequence. So maybe at this point over here, let me try something I can actually swap out. Maybe at this point over here, rather than A and a T, there is a G and a C instead. Largely everything else is the same. That's why it's still a protein for I. But this one change could have changed the color altogether. Right? So when I say a different version of the same gene, what I'm saying is that largely everything else is almost the same. Just maybe a small little change here and there changes it slightly. And that's why we see so much variation from our hair color to our skin color to our height to our eye color to our blood types. If I say you and I have the gene to make eye color, yes, that's true. But your gene may be the CG version. My gene may be the AT version. Okay? So you learn this in JH4. Okay, I might as well introduce the word to you. The word is called allele. If you ever see the word allele, it just means a different form of the same gene. So I'll show you how I use it in a sentence. I have the allele for black eye color. You have the allele for brown eye color. But it's the same gene. Understand the language? Yeah? You and I have the same gene. Okay, so I give you an example. Plants definitely don't even have this gene. Yeah? Unless you can find me a plant with eye color. But sometimes I try very hard to make you all laugh. I don't know what's funny about it. Yeah? And yes.
How does a protein like get change things? Like how does the protein like changing the sequence, right? Then how does this gene like send a message that like, oh, I want this color to be black? How does that thing work?
Right. Okay. I'm telling you, I have to oversimplify a lot of things so that we can appreciate the idea of genes and alleles, right? But very often, if I want to say what determines color and whatnot, usually it has to do with you change a particular enzyme in a certain way, then it may catalyze another reaction that result in another eye color. So I'm really oversimplifying so that we understand and appreciate alleles and genes at this point. Yes?
Is it possible to have human genes and plants create organs or
Okay, so see, now that you understand the idea of genes, then truly, if I want to create any protein, I just have to plug and play the recipe, put it somewhere else, right? Yeah? And that's what people have been doing. That's why we say genetic engineering. Yeah? People really just plug and play genes. And we've done it before. We do it with bacteria. So, for example, we take bacteria, and then we take a human gene, and then we put it in the bacteria. And then we create bacterial farms that create proteins that are human in nature. We have done it, and we are still doing it. We do it for a particular protein that's in short supply in the world, insulin. Okay, insulin is important to help regulate blood glucose sugar levels for people with diabetes. In the past, they used to harvest this insulin from pigs, right? And then after that, they inject it into human beings. But the insulin in pigs and the insulin in humans, right, are slightly different. And so it can cause a lot of illnesses. But ever since we discovered the concept of genes, then we've been able to plug this, put it into bacteria, get the bacteria to produce a human insulin. Have we done it for plants yet? No, we haven't. I suspect it's because plants don't have the same enzymes or cellular machinery. They can process the gene, the human version of the gene. But interestingly, the bacteria has the necessary machinery to process this gene, to interpret the recipe. Okay? So it's as good as I pass this recipe to an alien, which is the plant. If they cannot read the recipe, it's pointless. But the bacteria has actually the mechanisms to read our recipe. Yes?
Can you take this gene from someone else and put it into another human? So
Yeah,
like,
but some people are trying to do that.
oh.
So after all, right, let's say you have a particular genetic illness. If I want to plug and play, I want to put the healthy version of it and put it into you, can I do that? Technically I can. Okay, but you think about it, if someone has a genetic illness, this particular gene is likely found in every single cell of the body. So if you really want to effect any change, somehow you must get this gene into every single cell of the body. Now that is the real challenge. We found, scientists are finding it very challenging to do that. How in the world do you inject the gene into every single cell? So very often, if you want to make a change, you make the change right at the point before the being has even developed fully. From when a sperm just fuses with an egg and you are just but one cell, you make the change there. Right? Just change that gene one time, then when you develop into baby, all the cells will have changed. I see?
What happens if you do that but not for every cell?
What happens if you do that and what?
But not for every cell.
But not for every cell. Oh, then likely only that cell will have that change. Yeah? Only that cell has a different recipe. It will be a bit weird. Yeah? Okay, so to complete this process, okay, so we are very curious bunch. How do we convert this recipe into, at the end of the day, an amino acid sequence? There are two processes that we encounter when you play the game. The two processes are, number one, transcription. And then number two, translation. Notice the word translation. Translation is like you are converting from one language to another. That's what we're really doing. Okay, there's one exercise that we're going to try. But before that, let me just play this game first. Okay, we encountered this game last week. The idea of transcription and translation. To provide you some context, this is what you're about to see. If our genes are recipes, right, we need to somehow interpret this recipe and build the protein, right? Yes.
So if you have a cell and then it has like different cells, right? For example, your G cell and your blood cell, and they have the same type of DNA.
Oh, okay. Fascinating question. Okay, I ask you what you think, The cell at your eyeball and the cell at your nose, do they have the same genes? What do you think? Yeah? How about the cell at the eyeball and in your stomach? Do they have the same genes? Okay, in truth, every cell in our body has the same gene. Okay? Every cell. And then you think, that's mind-blowing. If the cell in my eyeball and the cell in my stomach and the cell in my toe has the same genes, right? How come my toe looks like a toe and my eyeball looks like an eyeball? Right? When I hazard a guess, how our body is able to... What will you do? What will you do if you are the body? Your cell here, here, and there have all of them the same genes. What will you do to make things different? Yes? Oh, say again. The first part was you said combine. Oh, okay. So how you suggest perhaps if I want to make an eyeball, maybe the cells over there will take segments of DNA that can result in an eyeball, right? Yeah, I'm not going to say no to you. There's one mechanism, but it is one mechanism. It is one mechanism. Okay, the more straightforward mechanism that our body uses, what you will learn that at university, what you just suggested. Wow. If only you're a scientist back in that day, so easy to discover new things, right? So I suggest, right, it's a new thing. Yeah, now, right, we just study more and more and more. There's more and more information. Every generation that goes by, we have more and more and more. Okay, so what does our cell do? We just switch on and switch off genes. Okay, just switch it on and off. So, for example, at our eyeball, if I don't need the toe gene, then I'll switch it off. Okay, and then my toe, I don't need the eyeball gene. I don't want the eyeball to stop there. Then I'll switch off the eyeball gene. Okay. Then your next question will be, how do we switch on and off stuff? There are various ways. One of the easiest ways is that we can attach things to the genes. If we attach something there, it may block the enzyme from going to it, then it cannot be read. That's one of the ways. Is
it like the allosteric site?
Not exactly. Different mechanism. Okay, so let's look at this particular process. Okay, wait, this is a very long-winded process. Let's watch two more. Okay, if you go to cellular structure and membranes, then we go to, what is this chapter? This is cell chapter. Why do I not find one? Oh, okay, I'm gonna play a quick summary video of the 2 steps to synthesize proteins. I want you to pay attention to what happens to our DNA. Sorry, I'm listening to lo-fi beats. Okay.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When the gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of 3 bases in the nucleus. The DNA code determines the order in which the 3 bases are added to the messenger RNA. This process is called transcription.
Okay, so that's the first step to produce a protein. Number 1, we need to make a copy of our recipe. Why make a copy? Okay, you know why? Because at the end of the day, if this is the library and this is the entire recipe book, doesn't make sense if you just want to make one small protein that you go and borrow the entire recipe book. Right? Just photocopy that page of the recipe book. Yeah? So that's essentially what our cell is doing. Rather than taking the entire DNA out, you just want this small little segment of the gene, let's just photocopy that gene. And then we can build whatever we want to build from that photocopy. Yeah? It also helps to protect our DNA. You know how when we borrow books, we will damage it, right? So don't lah. Let's not touch the original copy. Let's make a strong photocopy of what we need. If that one gets damaged, it's fine. Your main DNA is not damaged at the end of the day. Okay? So that's the first step. You need to make a copy.
Wait, Mr. Tan.
Yeah?
Why does it say 23 chromosomes? Like what's the significance of that number? Why is it only 23?
When they say 23 chromosomes, they are saying that in every cell in your body, there are 23 DNA molecules. Okay? There are 23. Sorry, there are 46 in total. 23 pairs of them. You will learn this in JH4 also. Okay, but just a very quick one. In your cells, there are 46 DNA molecules. Each DNA molecule has different genes. You have a lot of different genes.
What are chromosomes? Yeah, what are chromosomes?
Chromosomes are just DNA. They have been coiled up.
Before the messenger RNA can
Yeah, chromosomes is just a coiled form of DNA. Because your DNA is so incredibly long, we need a better, more efficient way of packing them into your tiny nucleus.
Why is it so
Then
like?
you know like it's not as a thing that attached to the start of the gene, right? So you mean it cuts into the DNA or like
Or it just opens up the DNA. So if you watch this part over here, you see this is, let's say this is a segment of your gene. Then you see this big enzyme coming in. Yeah. So the first step on transcription, purely, this enzyme comes in and spreads apart the gene so that you can create your mRNA. You need to open it up so that you can do your photocopying process. Yes? Oh, how does it know where to start? Okay, in university, you will learn that right before the gene, there is a special sequence that the enzyme can recognize, know that this is the start. Okay, there's a special sequence. And you will learn that special sequence when you go to university. Yes? Okay, how is it breaking it? It's just unwinding it and separating it temporarily. Yeah, after it's done, photocopying whatever it wants, then you'll coil it back.
Why are there like?
Right. Sorry, yes?
Why is it so specific? 46 DNA sequences in every cell.
Oh, why 46?
Yeah.
Why? Sorry?
Like why so specific? Like every cell has like is in different parts of your body. They won't need the same thing. Why all of them have exactly 46? Like why so specific?
Oh. Oh, I don't know. This is like I have to ask God. So okay, I don't know why every cell keeps everything. If I have to go back to the roots, it's really because we started off as a single cell. And when we started off as a single cell, we had all 46. We have every book, every recipe book we ever need in our original nucleus. And then when we divide and divide and divide, and then slowly become a multicellular organism, every cell starts to switch on and switch off different parts, different genes, and then different parts start to develop. I like to think it's because back in our evolutionary line, we were all okay, whether you believe it or not, we all started off as single cellular organisms, right? And every single gene in that single cell organism is important. Actually, all of it is switched on, right? But it's when we became multicellular that we needed to switch off some and switch on some. So I think it's because of the progression that we took from single cellular to multicellular, but we didn't quite change the aspect of removing genes or adding more genes. It's just it's more efficient to just switch on and off.
So every cell, all 46 DNA sequences are the exact same.
Yeah, exactly the same.
So there's only 46 DNA sequences in our
Correct.
entire body.
No, not in our entire body. In every single cell of our body.
Every single cell is in our entire body.
Right.
So every cell is identical to each other.
Every cell is genetically identical,
Oh.
but what proteins they produce can be very different.
So some switch on and some switch off.
Yeah. So that's how our body works.
hey, listen,
Yeah.
so when the two separate, right, additional bases will come in and then like pair to the complementary one, and then like after it goes through the big thing, the middle ones will come together, and then
Right.
that's the copy.
And that is correct. That's
But then
the
when the
mRNA.
middle ones come together, the big part, the top of the hammer is facing each other. Like normally it's the tips going like that, but then when like the complementary ones going, it's the opposite.
Oh, okay, wait, I play it. Then you see.
messenger RNA out of 3 bases in the nucleus.
Okay, so what are you saying?
No, but what about the ones on top? Like
Yeah,
the ones on top aren't gonna
The
be.
DNA.
Right, okay. So if you look at this, you notice that the enzyme is only concerned with one strand. The other strand, right, almost like useless, right? Okay, so there's a name to this particular strand. We call this strand the template strand. Okay, this strand is called the template. The strand on top is called the non-template. Okay. So this is the bottom is the template, there's a non-template, and we're using the template strand to make our copy. Yes. Right. How do you determine which is a template strand? Okay. Now we try our CA. Go to your CA, scroll down. Here I provided you a DNA sequence. Okay, here you see a DNA sequence double-stranded. Can I first get you to write down the template strand for this DNA sequence? You know how to do that? Okay, just write down the template strand. Or in other words, just write down the other DNA strand of a double-stranded DNA. Okay, everyone just do the template first, huh? Then we'll move on to the mRNA.
Does it work like the RNA, the red color one attaches to the green thing. So after you finish attaching, does the green thing like dislocate itself from the original strand and go out?
You're talking about does this red thing detach, is it?
No, does the green thing detach from its original to go with the red?
What green thing?
The bottom, the template. Like does it
Oh.
go together with the red? Otherwise it's just the one red.
Yeah.
Right, correct. It's really that one red that just comes out.
But it's no longer a copy because the copy is one on top and one below.
Yeah.
Right, okay. So I need to let you know, every time I see a copy, right, okay, you are highly sensitive and aware. Okay, so notice, right, when I say a copy, it's a complementary copy, right? It's not really a copy of the DNA sequence, right? A true copy would be, I need to copy both the top and bottom. I don't. There's only one strand that is important. I'm actually only copying, and if you notice so far, you can rewatch the video. Every time I say copy, I put air quotes. I'm not exactly copying, I'm complementary copying.
So technically he's copying only the non-template one.
It's copy template.
In a sense, okay, you notice I'm complementary copying the template, and you notice the sequence is very similar to the non-template. So that's why every time I say the word copy, I put this up. Okay, have you written a template strand? This is something that you should have been able to do from your topic of DNA.
Oh, I know already.
Shall we run through the sequence? Okay, I just do the first three, then the rest you should be able to do. T, A, C, then followed by G, G, T, C, C, G, C, C, G, C, T, C, so on and so forth. Okay?
Oh, the complementary.
Right, okay, now, whatever we just watched in the video, that process is called transcription. Okay, now let's try the next skill. Can you transcribe an mRNA strand from the template?
Oh, the mRNA is just a non-template.
Oh no, because RNA,
just
the bases that complement each other are different from DNA.
Okay, I really don't overcomplicate this. It's really quite a simple skill. Okay? Can you transcribe the mRNA strand from the template? When I first learned this, one question popped into my mind. Then what in the world is the point of the non-template strand? It's like useless. We might as well just have a single strand of DNA. Why have double stranded? We are doing the okay, everyone try. Can you write down the mRNA strand when you transcribe the template strand? The skill to use here is complementary base pairing.
Okay, so because RNA, like the bases are AT, A, U, and C,
Right.
and DNA is AT and C, so does that mean that mRNA is not exactly identical?
Exactly, it's also even not. Yeah, so every T that's supposed to be here is a U.
Is that the only difference?
Oh.
Yeah.
So that's what you mean when it says copying.
That's why I keep saying copy.
Copying in its own version.
Okay, are you ready? You managed to transcribe the mRNA. There's actually a cheap way to do this. Really fast. If you figure out the fast way. The most straightforward way is you really go by the book. You whatever sequence was written here, let's say it was TAC, right? Then you write the complement of it. A, but rather than a T, you put a U and a G. Okay, what's the shortcut way? Okay, copy the non-template strand down, swap out all the T's with U's. Okay, that is the fast way once you realize the commonalities. Okay? Okay, so what you just done is called transcription. I take the template strand and I create a sequence. Oh shucks, I need to think up. U, thank you. C, 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 G, C, and A. Okay? And I create an mRNA. This is transcription. Okay, so I don't want you to feel overwhelmed by all the enzymes involved and whatnot. You focus instead on the skill. The skill here is to be able to make a copy. Okay? So what can I do? I may provide you a new sequence, then I'll ask you to transcribe it. That's all. The next part is a more exciting part. Now, how do we create an amino acid sequence from this recipe? Okay, once I'm done with my DNA, this is what our cell will do. It will then close up the DNA. Okay, close up DNA. I do not need my book anymore, so I'm going to close the pages. Very good. Now I have my mRNA. Okay, the next skill to have, you need to refer to your textbook on this particular page. You go to lesson notes. I want you to go to this particular table on page what? Wrong set of notes. This set of notes. It's called genetic code. Over here, page 24. Okay. Okay. To pick up this second skill is to be able to read, to make use of this thing called a genetic code. Okay. Do you have to memorize what each triplet represents? No. Okay? Don't need to. Instead, if we want you to perform the second step, translation, we will provide you this genetic code. Think of it as the periodic table in biology. Yeah? Okay, so what is the skill here? Okay, I'm going to tell you this skill is not very difficult at all. Okay, you look at your mRNA sequence. How our cell is able to interpret this recipe is by reading it in triplets. So you see here, this represents one amino acid. This represents another amino acid. This represents another amino acid. So the skill here really is to, can you make use of this decoder to help you interpret what the mRNA is trying to say? Can you try now? You try for my sequence over here. Glycine, is it? Proline plus. What's GCA? Is that it? Yeah? Okay, then that's all. This is the second skill to be able to pick up that you can decode. Okay? So the first skill is to transcribe, which is a complementary copy. The second skill to pick up today is just to be able to decode. Okay? Yeah. So now you try now. Okay. Sorry, let's see what you asked.
What's the point of translation?
Okay, what's the point of translation? Do you see what I've just done here?
Yeah.
I've created a primary sequence for a protein. And remember we said that transcription and translation is the process to synthesize any protein you can find in your body. So what I've just done here is that I've just translated this language into the amino acid sequence. I've created a primary structure. And all of these are amino acids, by the way. Okay, sorry, if I'm not clear, everything you see here is an amino acid. Yeah? Okay?
All these are amino acids.
Right, yeah. These are names of amino acids. If you don't believe, you go back to your chapter on proteins, you go to the table of amino acids, you'll find exactly the same name.
Isn't that R group?
Yeah,
no, no.
R groups gives you the amino acids.
It's R group of amino acid.
Those
Okay.
are R group.
You go, really, you don't believe, you go to the chapter on proteins, search for the table of all the amino acids. It is the same amino acids you find over here. So, can you try this second skill now?
Those are amino acids. Yeah, this is the.
May I get you to translate your mRNA? Okay, can you translate your mRNA now? Okay, that's the second skill to pick up today. Yeah. Where do we? Oh, okay. Write this space there.
That is the name of
Right.
the
Okay, so
amino
you
acid.
see the start folder? That's actually an amino acid. It's called so that is an amino acid. ME. Like when you stop, does that amino acid, right? So when the enzyme, the ribosome encounters the stop, then you stop it. It's like a full stop process. Right. Right. Okay. Just translate and write the amino acid in these boxes. Okay, then we'll do a quick check with each other if you translated correctly. Yes, okay? You can write the short forms. For example, GLY, that's universally accepted. When you write GLY, I know it's glycine. I know that's choline. I know that's alanine. It's like a periodic table, really. Yeah? You don't have to write the full name of a particular element. O represents oxygen, so on and so
Does
forth.
that even include like in test includers?
Say again.
In test includers, is it also the same thing? Or do you want us to like write out the
Oh,
Yeah.
in tests and quizzes, can you write out the amino acid name in full?
Okay.
But we'll provide the genetic code. Don't go and memorize this code.
Yeah, I need that. And can I fix up the rest
Right, okay. Now, I need to answer the question. Okay, how do you know which is a template, which is a non-template? Okay, your template strand very often will provide it to you this way. Notice that one strand is 5 to 3, one is 3 to 5. One way to tell which is a template strand, go and search for the one that is 3 to 5. Okay, that's one way. Okay, your mRNA. Okay, then you add. Okay, I know, right? Something's on mind at the moment. Okay, so one way you can decide which is going to be the template strand is to go and look at which is starts with a 3 ends with a 5. The mRNA that you created, okay, it actually runs in a 5 to 3 direction. But it's also anti-parallel when you create an mRNA. It's also in the opposite direction. Next question you probably ask is, wait, if I say that, take the one that's 3 to 5, if I just take this entire DNA sequence and I take it topsy-turvy, then wouldn't the other strand become the template now? Right? Do you understand what I just asked? Sorry, Zoe? Because the question is, how do you identify the template strand? Right? To identify, you look for the tree, the one that goes from 3 to 5. This. Yeah, Isabel? Alright, Matt. Oh, start codon is just a name for the very first amino acid that you will put in. Okay, so put in MET. I should see that all of your proteins, all of your primary sequence should start with MET. Or rather, does it stop codon and amino acid? It doesn't count. See? Like, why are they like listening or two? Right, okay. Can you remind me if I don't address this? I'll address it in a while later. Okay? Okay, have you translated? Once you've translated, check with your friend if you translated correctly. It's purely about being meticulous, being able to read the genetic code. Yes, Kaisi? Oh, like can you combine genes to make a single gene? Alright, okay, whatever I've just done here, right? You'll never see this. Genes are much way longer in sequence. They are usually hundreds and thousands of.
You read them like 3 at a time, right?
Right, okay.
Then other organisms can read like 4 or 5 at a time.
Oh, okay. Are you saying that we are reading this sequence in triplets, right? Are you asking, are there organisms that read it in forms? No. Okay? Every organism you find on Earth, I say on Earth, but I cannot say on other planets, I'm not very sure. But every organism on Earth reads the genetic code in exactly the same way. And that is crazy. Because that really means I can plug and play some plant gene and put it myself also can. Because every organism in the world reads the genetic code in exactly the same way.
Then where do like there's some organisms that come in millions, right? Then where do they come in?
Oh, that one you probably have to put it up.
Yeah, like where he does, where's come from?
No, oh, but we're not producing these amino acids though.
Yeah, but someone else is producing.
Where did it come from? I'm not sure. Likely there was an enzyme that made it. But then where did the enzyme come from? I also don't know that. Maybe some are spontaneously formed. Sorry, Shanice?
What does stop codon mean? I think you can start.
Okay, let me write this down. I want to address it all together. Stop and start. Stop, start. Okay, have you all translated? Check with each other. Okay, let me show you my translated sequence. You have the same thing? Okay, what you've just done is just on paper. I want to show you in video biologically what this looks like. Okay? You must appreciate what it really looks like, not just be able to do the paper skills. This is what the process looks like in our body. Okay, after you done transcription.
Determines the order in which the free bases are added to the messenger RNA. This process is called transcription. The production of pre and messenger RNA then moves out of the nucleus into the cytoplasm.
Okay, there goes our mRNA. The mRNA is now in our cytoplasm. Okay, then comes the next step, translation. You just have to do whatever you've done on the paper, but appreciate this is what happens in the body. This is how we translate. Okay?
The cytoplasm, called ribosomes, bind.
This is where ribosomes come in. Okay? Watch what the ribosomes do. They do something quite interesting.
To the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid.
So what's actually happening is, the ribosomes need a little bit of help. They need delivery men. The delivery men are those plus-shaped things you see. They are called tRNA. They bring in the correct amino acids based on what this code is. Yeah?
This is added to a growing chain of amino acids.
Okay, you'll learn more about this in SH, but for us, the skill we need to have is to just figure out what's the sequence in the protein. That's all.
Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay? So a question has been asked. You notice that in the genetic code, there are these things called start and stop codons. You saw? Okay. What is a start codon and what is a stop codon? You notice that the start codon is the amino acid methionine. Okay? So, you saw that there are ribosomes just now, right? Ribosomes. Ribosomes need to detect methionine to be able to translate it. Think of it as wow, what to think of? I was going to think of an analogy here. Okay. Ribosomes just need to be able to detect this. Once it detects methionine, then it can start the translation process. You also saw that there were 3 stop codons. Think of the stop codons as full stops. The ribosome need to know when to stop translating. So, when it encounters a stop codon, can you give me an example of a stop codon from your genetic code? UAA, is it? So let's say the ribosome comes across UAA. You notice that the stop codons do not have any amino acid. It's really a full stop. When the ribosomes encounter a stop codon, it will just shit out the protein. Okay, it will stop the translation process. Okay? So for every protein that's synthesized, we need a start and we need a stop. Just to help us know when to start, when to stop. Yes? Oh, meaning? Oh, is there a tRNA for stop codons? If I'm not wrong, there is a tRNA, but the tRNA doesn't come with amino acid. No, it just comes in and then it signals the ribosome, hey, you can stop already, we are done. Sorry? Where do you find tRNA? Is it? They are just floating around the cell, attaching to new amino acids that come in and then bringing it to the ribosome. For us, the skill we need to have is to be able to transcribe, translate. Are you able to transcribe and translate? If you can do that today, then you are quite settled for this segment. Yes?
What happens if like the mRNA is like the stop codon triplet like in the middle of the mRNA, then will it form two different proteins?
Oh, okay. So, Christy, are you asking? Let's say we have an mRNA and then the stop codon suddenly, there's one stop codon right in the middle. Will you produce two polypeptides? Okay, if you suddenly got one stop codon right in the middle of the mRNA, which you will not find, if we suddenly there's one in the middle, you mean that everything else doesn't get translated. It means your full stop came early. Okay? So your mRNA, the full stop is right at the end all the time. Okay? You shouldn't find a full stop, the stop codon, in the middle.
Then like how does the mRNA know where to stop like the transcription? Then like so that there won't be a stop codon in the middle. Like does the stop codon also like mean that the mRNA will not transcribe the rest?
Oh. Okay, then what determines where the stop codon will be? It determines by what was the template strand, right? Okay, so for every gene, you'll find that there will always be a start and stop at appropriate areas. There shouldn't be a stop that appears too early or too late.
I mean like for the transcription, because the DNA is like very long, right? And the mRNA only takes a bit. So how does it know where to stop?
Oh, transcription process, is it? Oh, okay. Oh, okay, I understand. Ribosomes make reference to the start and stop codon to know when to start and stop, right? But how do the enzyme over here know when to start and stop transcription? That's your question, right? Technically, I could just transcribe a little bit more than the stop codon. Okay, so how does the enzyme tell? There are sequences before and after for the enzyme to tell when to start and stop. Okay, in university you learn that right before the gene there's a sequence called the promoter. Right after the gene there's a sequence called the terminator. The robot, the terminator. So enzymes recognize promoters and terminators to know when to stop transcription and when to start. Okay. Yeah. Okay, so that's one skill. Yes. Our DNA what? Correct, yeah? So I will just ask, so technically, does our DNA create amino acids? It doesn't. Our DNA is purely a recipe for ribosomes to stitch together the amino acids we want. Where do you think they come from? Yeah, from the food that we eat. Right, then your body just cannot make that protein now. So you see how important a balanced diet is. Yeah, if you don't have a balanced diet, your body may be short of a particular amino acid, then it cannot create this protein, even though it has a recipe book. Right? It's just like how you have a recipe, right, when you got no egg. You cannot just substitute the egg with something else. I tried before, it didn't work out. Okay? I told you before, I made a cookie, right? Substituted a chocolate bar with Nutella. My cookie just did not come out as a cookie. Yes? Oh, tRNA is really like a delivery man. Deliver already, then go around and find the next amino acid to attach to come back. Yeah, it can be recycled. Is the structure. The structure of tRNA allows it to attach to the amino acid in a particular. Yeah. Yes, yeah. Oh, okay. So let me interpret Sion's question. Are you asking for one mRNA sequence? Can you reuse it? Okay. Can you reuse the mRNA sequence? And okay, what do I mean by that? Imagine this is my mRNA. I have one ribosome. If it reads this mRNA once, then you ship out one protein. You read it again, you ship out another protein. So what does this mean? Your mRNA is recyclable. Depending on how many proteins you want to make from that one mRNA, you just got to run through how many ribosomes that you want. In fact, I can show you one image. They found mRNA ribosomes micrograph. I've seen the image before where one mRNA has multiple ribosomes attached, running like a choo-choo train. Each one going along the mRNA, and then you can create many proteins at one go. Okay, this strand that you see here, that's mRNA. Do you see all these dots? Those are ribosomes. This mRNA has so many ribosomes running along it. You know what this means? That from that single mRNA, I can actually produce many of the same proteins. It is as good as I have one recipe here, then I have 5 chefs surrounding the recipe, then we cook it together. We make the same thing together, you know? Maybe that's what we can do. Yes? Oh, okay, fair. Okay, then what's the alternative? Right, okay. But then what if you only need the enzyme or the protein in a particular scenario? Right? So our body can regulate the creation of proteins or depending on whether we create an mRNA. No. Actually, this relates to switching things on and off. Yeah? So, in our eyeball, if we do not want the TOW gene to be transcribed and translated, then what do? We make sure that no ribosomes can attach to it. We make sure that transcription doesn't happen. We make sure translation doesn't happen. And that's how we can switch things on and off. If we want the eyeball gene to be transcribed and translated, then we want to make it available. We want to let the enzymes reach it, to transcribe it. We want to throw a lot of ribosomes to the mRNA so that we can create all the proteins required for the eyeball. So that would be the purpose of why we want to even create mRNA. Yes? What if the ribosomes catch up to one another? That's a great question. I don't know. Maybe they were like bumping each other, maybe they'll stop. I don't foresee a big issue. Yeah, I don't think that will be a big issue. Interesting question. I don't know actually. Yes, you should. Yes. Oh, okay. You should ask, if genes are the cause for genetic diseases, why don't we just switch off these genes? But what if this gene is required to produce something that's important in your body? For example, what if you have a genetic disease related to your blood? If we switch off that gene, then you don't even have blood. Right? So it's not that easy to just switch things on and off as we want, because sometimes this one gene may be in control of many things. You cannot just switch off one part of the gene. Usually, you want to switch off, you switch off the entire gene, or switch on the entire gene. Okay, in preparation for our next lesson on Friday, I'd like you to prepare something up. Okay, number 1, let's answer this question. Okay, compare and contrast the non-template DNA strand and the mRNA strand. Okay, I think by now you noticed that the non-template strand and the mRNA, they are almost the same in sequence, just that you swap out the T's for the U's. Yeah, so that's the first thing you want to compare. Can you fill in the blanks for B? During transcription, RNA polymerase, which is the enzyme that transcribes the mRNA, what bonds are being used to produce the mRNA? Okay, I hope you can answer this yourself. Then during translation, when the ribosomes are linking the amino acids together, what bonds are being formed between the amino acids? Okay, I'll leave these two to you to ask to discuss among each other. Sorry, Kada, you have a question? Okay. Polymerase is an enzyme that creates a polymer of RNA nucleotides. Okay, so polymerase. Or I simplify it up. You know what polymer is? Yeah, polymerase just creates a polymer. You want to see a rat that has a human ear? I think I've seen it before. Like, yeah, this is what happens if you go and put a human gene on a rat and then you switch it on. Wow, what the hell? Rat growing. It's quite nasty, huh? What is that? Okay, so the question is, now that we can, should we? No, I have no idea whether it can or not. Yeah, think about it, huh? Technically, if we can just modify our genes to create anything that we want, then doesn't this open the door to creating the perfect baby? No. I mean perfect in your eyes, in society. You go to the hospital and say, I want a baby with blue eyes, then you select this gene and then gene and gene together. Yeah, right? Yeah. So should you? Even if we can. Sorry? Yeah, and you need a central bank of all the genes. Would you want to? I don't know, maybe there's some, I don't know, I imagine as parents, there's this joy, right? There's this like, it's like you play those, you know, you ever play those like slot machine, you turn and then one ball come out. I always thought having children is like that, you know? Like you just put a coin, right? Then you turn, then you hope for the best. Yeah. I'd rather that, right? Or alternatively is that you get to choose out of a catalog exactly what you want to do come out. Every time I talk until this stage, I forget what to do. Okay, so number C, or rather alphabet C, transcription is sometimes described loosely as photocopying of the DNA. To what extent is this true? I think by now we've had enough discussion to realize that it's not exactly a photocopy. Okay? Anyway, all these questions are just questions to get you to think. Huh? Yes. What? Okay, if you turn a rat into a human, is it a human or a rat? If you turn a rat into a human, what makes a human a human then? What makes a rat a rat? If I clone you, I transfer all your memories to this body, are you still Yishuan? Okay, to prep for the next lesson, this is what I'd like you to do. The reality is that what I just painted to you is the most beautiful scenario. It's as though our body does not make any mistakes. Heck, if I ask you to copy something from a whiteboard, you cannot copy it correctly word for word. Right? So, in reality, our body makes a lot of mistakes all the time. Our genes very often, they are not maintained as perfectly as we hope for it to be. Sometimes mistakes happen. I'm going to show you 3 kinds of errors, changes that can happen. No, hey, sorry. Nothing to do with parallax error. Okay, so I'm going to show you 3 kinds of mutations that can occur. Not 3 kinds of mutations. 3 changes that can happen to our genes that can be pretty serious. Number 1, substitution. Sometimes our body makes certain errors, and the errors can go like that. If our gene is supposed to run in this sequence, sometimes some error occurs, and then we swap out a pair for another pair. Yeah, come to a point in a while. This is quite serious, right? You think about it, what are the implications? If I substitute this out, downstream, going to create a different amino acid. Yeah? Okay, so that's the first kind of change that our cells may accidentally make. Second kind of change, okay, I want to put this back up. The second kind of change goes like that. Sometimes our cell may accidentally include one more pair in the recipe. So, for example, I may accidentally insert something in between. So you see what happens? Originally, the ribosomes were reading everything in triplets like that. If you go and insert one more over here, let's squeeze one of them. Notice my reading frame shifts. Okay, if I were to insert, let me insert a new one over here. Up. You see what happens when I insert? Now I read everything slightly differently. This is the first triplet now. This is the second triplet. Everything from the second triplet down gets displaced. Right? So this is another serious change that our cells are quite capable of doing. The last change is this. A deletion. Sometimes this may happen. Instead of inserting, I accidentally delete a pair, then your frame of reading gets shifted upwards. Oh, then you're missing something. Okay? Yes?
Then what happens to the stop codon if everything gets moved?
Okay, we will see what happens on Friday, okay, when you do all these changes. So this is what I'd like you to do. Do I have a lesson you do on Friday?
Yes.
Okay, so this is your task, this is your homework. I just introduced to you 3 changes that can happen in our body, in our genes. Substitution, insertion, and deletion. Okay, now I want you to train your eyes to spot the changes. Go to your CA. Okay, look at this page over here. I provided to you the original non-template strand. I did not provide you the template strand. Okay, just save space. Because you know that the non-template strand is essentially the same as the mRNA. Okay, so you can try this now, but by next lesson we will have a discussion. Can you try to spot the change that has happened? Look at table 2, 3, and 4. Can you tell what the difference is compared to table 1? That's the original gene. Can you tell at what point I substituted? At what point did I insert? At what point did I delete? That is the skill to pick up. You need to train yourself with a very sharp eye to spot where we induce the change. Can you? I only made one change to every gene. Are you sharp enough to spot the change?
You mean the change in the same box, is it?
What do you mean by that?
Like, is the second triplet or like that you made on, is it only one triplet? Like, do you only make one change throughout the whole strand, or do you make?
Oh, okay. I only make one change. Okay, I only make one change. So I either substituted one thing, or I inserted one thing, or I deleted one thing. Are you able to spot it with your sharp eyes? You spotted already? What did I add? What did I add?
Okay, then I'll make sure that I'll borrow it.
Okay, so after you spotted the change as your homework, Can I get you to translate all of the sequences? Can you translate all of them? There are 4 rows to translate. Can you practice your using the genetic code to translate? On Friday we have a discussion. How do these 3 changes affect translation? Okay? So in your free time now, you can translate all the sequences and then you can compare to the original one. How serious are the outcomes? Which one is most serious? Substitution or deletion or insertion? Yeah, next lesson we'll have a look at what you translated and then we'll see how serious the problem is when we substitute, delete or insert.
Then how does your body like react to these changes?
Right, okay, so when we are still young, we have proofreading mechanisms in our cell to make sure that things are still correct. But as we grow older, these proofreading mechanisms start to fail. That's when we start to develop cancers. Right?
Why would it make these mistakes though? Why would it?
You know our cells divide, right?
Yeah.
Every time our cells divide, it needs to make a new copy of the DNA.
Yeah.
When it's copying the DNA, that's when it can make these mistakes. Yeah. So imagine I ask you, I want to construct a new library, but I need to go and create a new copy of every single book in this library. Some errors may be made when you are trying to create a new library on in the other cell.
Wow.
Are your practice sets inside these things? Or they are separate? Are they digital or are they hard copy? So these are not practice sets. Then what is this? Oh, this one you do in class. Right. All these are class activities. Oh. Oh, I see. What's the QR code for? Yeah, there are a lot of QR codes. What, volume 3? So you have 2 volumes already. Right. Okay, you all may go out for your break. So on Friday, we will have a look at what you translated. So, see you in the IS lab at 2:30, and we try and at 4 or earlier.